as I say, this might be a bit of a rehash. I'm just going to quickly refresh your memory. So we've got, obviously, there's a host of different probiotics on the market, as I'm sure you know. There's, you know, really it's one of the most, you know, there really are hundreds and hundreds available globally. And Australia, as you know, in Australia, we don't need the product registered. So like in the US, it's generally recognized as safe. So as such, you know, there are many, many products on the market. We can sort of classify them into three types. The spore formers, which is the group that Correlink falls into. Of course, these tend to pass straight through the gut of the organism. They have their action in the gut, but then they passed out. They don't actually colonize the gut as such. So this is one of the reasons why Correlink is fed from day one all the way through to end of life, because it's not like, for instance, some of the live vegetative forms where they actually colonize the gut, and generally you only give them in the beginning and then they establish themselves. So that gives you a quick overview just to understand what the difference is there. And then, of course, there's some products that only, where you don't actually feed the microbial as such. You only, you feed the product of the microbial, so the lactic acids or, you know, things that the bacteria would produce, rather than feeding the bacterial spore like we do with Correlink. Yes, so Correlink is actually a Bacillus subtilis, and you would have seen on the market there are a number of Bacillus subtilis that are available. We'll be talking more about what are the differences between these in terms of, you know, why do we have 6 different strains of Correlink, and when it's, you know, exactly the same Bacillus subtilis, what is the difference? So we'll talk a bit more about that. I have to touch on what a bacteriocin is, because this is really key in understanding why we have the 6 strains of Correlink and why we customize it to each customer. So really the action of probiotics and how they would work is that each of the bacteria that we feed would be producing an array of peptides or proteins. We call them bacteriocins, and those are actually the key molecules that actually have the effect on the gut in different ways. So if we look at it visually quickly, there are a number of ways in which they benefit the gut. So the first way is these bacteriocins. If you look at the first diagram here, the blue is your probiotic that is being fed. They produce these little red dots, which are the bacteriocins. Now these can facilitate the probiotics to actually establish themselves in the gut by making the environment more beneficial towards them. The second way here is that these bacteriocins can actually directly affect pathogens in the gut, so sort of have a killing action directly in the gut. The third way is that bacteriocins can signal to the host immune system, and these they will signal to the host to actually activate the immune system, which then has an effect on pathogens in the gut. So we are obviously very interested in what bacteriocins each of the probiotic strains are producing. All right, so just quickly, are all Bacillus species equal? So no, obviously these are strain specific. But then if we look at Bacillus subtilis, you know, why are we saying Correlink is different to other strains? Or why, you know, if you're feeding a bag of Correlink, why is it different to feeding a bag of, you know, a different company's Bacillus subtilis? Well, it all comes down to what strain you are feeding. So there are many, many different strains of Bacillus subtilis. We focused in on 6 strains. We used to have 7. 1 was replaced, so we're constantly evaluating sort of what are the best strains globally to use. And each of those strains would produce a different array of bacteriocins. So that is the key there, and that is why we try and customize the Correlink strains that we are using per customer. We'll get to that in a minute. So it's not, you know, each strain, if you're feeding a Bacillus subtilis, it's not going to be the same bacteriocins being produced if you're feeding 2 different strains. All right, so surely then if we're feeding more strains, if we, why don't we put all 6 of our Correlink strains into the feed? Generally speaking, obviously it would be great to have that diversity and have all the bacteriocins, but you've got to remember then those will be diluted in the feed. So that is why we usually focus on 2. And you'll see by your report that that's, more than adequate. I think we've got 1 customer globally that has 3 strains in their feed. It all depends on what results you get out of the sampling.
Lindy,
and of course, it wouldn't be. Yeah.
Are there any prebiotics that are also included in the Correlink to assist the production of the bacteriocins?
No, not at all. It really is just the raw bacterial strains that go in.
Raw bacillus, yep.
Yeah.
Thank you.
Okay, so that was a very brief background. We'll now go into Correlink and how the sampling part works. Here you can see the 6 strains lined up at the bottom. So really it's a custom blend out of 6 potential strains. And the key for Correlink is that we are focusing on 2 disease conditions. We're focusing on avian pathogenic E. coli and toxigenic Clostridium perfringens in the gut. And our measurement is basically a measure of the level of each of those 2 bacteria in the gut. So just looking at each one individually, if we're talking about pathogenic E. coli, obviously there are many colis in the, in the normal healthy gut of a bird as well. So not all of them are classified as pathogenic or disease causing. So it's important just to note that, because you'll see in the Correlink report, there's you get a graph showing the overall level of E. coli, and then out of that you see which of those are actually pathogenic. Now, when we talk about pathogenic E. coli, these would be the E. coli that cause the generalized colibacillosis syndrome in birds. Obviously, fibrinous lesions around visceral organs is one of the most common, sort of disease states seen in broilers. So I'm sure you're familiar with those. Symptoms, asacculitis, cellulitis, etc. So not so much a gut, the symptoms aren't always visible in the gut, but the bacteria originate in the gut. So that is why, so it's just important to see that link, that if we are decreasing pathogenic E. coli in the gut, we're not necessarily going to be affecting the gut health. We're affecting the syndrome of colibacillosis in those flocks. Now, just quickly, if to understand how does the laboratory distinguish between the normal E. coli that are isolated out of the broiler guts and the pathogenic E. coli. There's a very good paper by Johnson et al., which I can share with you as well, and they talk about these virulence factors, which are encoded by genes in the E. coli organisms themselves. And there are 5 genes that specifically give these E. coli, should I say, more sort of the superpowers to become pathogenic, and they'll code for things like receptor genes or hemolysin genes, things to help them survive in the host and to cause disease in the host. So for instance, you can see serum survival genes that will enable better survival in the host, hemolysin that'll cause hemolysis of red blood cells. So any of the pathogenic E. coli that are isolated from the broilers at Hazeldene's, if their DNA contains 2 or more of these 5 genes, they are immediately classified as pathogenic. All right, so that's how we classify it. We'll, even in the report, you can see further down, we will classify what percentage of the pathogenic E. coli out of the broilers had 2 pathogenic genes, how many had 3, how many had 4, how many had 5. So you immediately get a visual sense of, you know, what is the sort of virulent status of the E. coli that are being isolated from the broilers. So that is where that data are being drawn from. Clostridium is much simpler. Basically, if they have the alpha toxin gene, then they are obviously picked up as toxigenic Clostridium, causing necrotic, or with the capability to cause necrotic enteritis. We have in the past isolated, well, actually not in the Correlink samples themselves, but I know there have been farms where the beta toxin has been present. So the lab in the US is able to test for both of these, but actually in this